Good morning to everyone. Today we are going to discuss about the topic on DNA replication. I am Dr. Krishnakumar, Assistant Professor, Department of Botany, Cordon Planter Association College. Let's move on to the topic. General introduction about the DNA replication. Replication is a copying of the DNA. DNA has ability for the self-duplication. DNA replication occurs in all living organisms from prokaryote to the eukaryotes. The mechanism of replication from bacteria to the humans. Replication occurs in single strand and the double strand DNA. Replication to the parent strands function as a template. Newly synthesized strands is a complementary to the parental strands. DNA replication is a semi-conservative process because in one strand two strands is produced. One strand is a parental strand and other strand is newly synthesized strand. Replication is a semi-discontinuous because in one strand of the DNA is synthesized in a short fragment, in other strands it is synthesized continuously. Replication in both directions because it is opposite direction in the two strands. Replication involves three steps, namely initiation, elongation, termination. During the replication, deoxyribonucleotides are added one by one, resulting in the formation of polynucleotide chains. The formation of the polypeptide chain by addition of nucleotides is called polymerization. The addition of nucleotide is catalyzed by the enzyme DNA polymerase. The synthesis occurs always 5' to 3' direction. Now move on to the topic, mechanism of DNA replication. The DNA replication has extensive studied in the E. coli, in the phage and the plasmids of E. coli. The process of DNA replication involves three steps, initiation, elongation and termination. Let's move on to the topic, initiation. Initiation comprises three steps, recognition of origin, opening of duplex to generate the region of single strand DNA, capture of DNA P proteins. The DNA A and ATP complex bind at the 9 base pair inverted regions of OIC of E. coli, promotes opening of DNA duplex in the region of 3 direct repeats of 13 base pair sequences. It is called 13 mers. Opening occur from right 13 mers leftwards and require the natively supercoiled DNA, HiQ or inhibitor proteins. So this is the overall picture about the DNA replication. So now here we can see the double strand DNA. It is unwinded by the DNA helicase enzyme, and then there are two strands, one is leading strands and lagging strands. Leading strand is a continuous strand, lagging strand is a discontinuous strand. This discontinuous strand is called as a Okazaki fragments. Let's see detail. DNA P helicase is transferred to exposed single strand DNA and cause unwinding of DNA in the presence of ATP, SSP protein and DNA guidase. This results unwinding of DNA duplex and the replication from OIC process in both directions. SSP occur in a single strand region and the two DNA P complex are loaded on each strands. Next, move on to the topic, elongation of DNA chain. These steps involve the process of enzymes, DNA B helicase, primase, DNA polymerase holoenzyme, SSP protein, RNase H, and remove the RNA primers. DNA polymerase 1 is used to fill the gap created due to the RNA primers. And finally, DNA ligase, which convert the primerless Okazaki fragment into continuous strands. During the initiation to the elongation transition, the following steps are involved. As a helicase travels from 5' to the 3' direction, it generates replication forks by the opening of DNA duplex. The DNA strands having the helicase because the lagging strands of the DNA primers are associated with the DNA helicase, forming the primers which synthesize the multiple primers for the lagging strands. Single primer for the leading strands. For the synthesis of lagging strand, DNA polymerase III holoenzyme to work in the same direction to which DNA B helicase is bound but travel in opposite direction. DNA helicase and DNA G primase and DNA polymerase III work together in a strand elongation. Helicase and the DNA polymerase assembly remain processive. They remain tightly bound throughout the reaction. Elongation of the lagging strands takes place by the somewhat different method. It is far more complex for the lagging strands for the leading strands. Sorting by lagging strands, the discontinuous synthesis of the lagging strand. The primase which is taken up from the solution is activated by the helicase B to synthesize the RNA primer on the lagging strands. The RNA primer is recognized by the DNA polymerase III holoenzyme on the lagging strands are utilized for the synthesis of precursor or Okazaki fragments. In fact, each RNA primer is recognized by the gamma subunit of DNA polymerase III and loaded with the B subunit of the same prime polymerase. This preloaded P subunit may capture the core of DNA polymerase III enzymes when it becomes available after finishing the synthesis of job on proceeding the Okazaki fragments. On the complexion of Okazaki fragments, the RNA primer are excited by the DNA polymerase I, which fill the resulting gap with the DNA. After the DNA polymerase I is added to the final deoxyribonucleotide in the gap left by the existing primer, the enzymes of the DNA ligase form phosphodiester bond that link the free 3' end and replace the 5' end of the Okazaki fragments. Next one, continuous strands on leading strands. In both direction DNA replication, leading strands is a prime once on each of the parallel strands. RNA primers of the leading strand is synthesized by the RNA polymerase enzyme. DNA polymerase III causes the elongation of the leading strands by finally DNA polymerase I and ligase enzymes give the final touch to the leading strands in the case of lagging strands. So final one, termination. After the two strands synthesized from the parallel strands, new strands form and join with the new duplicates of standard DNA. The parental strands rejoin as a double strand DNA. This process repeats again and again to form multiple replication of the DNA. So that's all about the DNA replication. Thank you.